Okay, so welcome to today's astrobiology seminar. Today's speaker is Furkan Ozturk. He grew up on the southeast shore of the Black Sea, did a degree in physics. He did an internship, a couple of internships. One was at Indiana University in physics, one at ETH Zurich. And then he went to Harvard. And he's been doing a PhD there, and he's basically finished apart from his final exam, which is just over a week's time. And he's been working, he sort of started on atomic, molecular, and optical physics, and then somehow found the origin of life. So he went from the dark side to the light and started working on something relevant to astrobiology. And he's published a few papers now on his new ideas about producing homochiral molecules in the origin of life. And that's why I've invited him, because he's kind of at the cutting edge of that research. So take it away.
Well, thank you so much for the introduction and for the invite. And as David said, today I will talk about the chirality problem of the origin of life and how we address this problem. So I will start by introducing the problem, and then I will talk about an effect which was discovered kind of recently that can be very instrumental in addressing the chirality problem. By the way, can everyone hear me in the back? No mics needed? Good, okay.
Speak loudly.
All right, and then I'll talk about the experiments that we did to basically utilizing that effect. And then last bits are going to be about how to realize a homochiral network as opposed to just making one compound homochiral. So, well, what is biological homochirality and what is chirality to begin with? Well, chirality is a geometrical property, and objects are said to be chiral if their mirror images are not superimposable on each other, just like our hands. And biomolecules, most of them are chiral as well. And amino acids and nucleic acids, sugars, peptides, they're all chiral molecules, and they can exist in near symmetrical pairs. As you see here, for instance, for ribose and alanine. The interesting thing is biological systems are extremely picky when it comes to chirality. In biology, we exclusively, with few exceptions, see left-handed amino acids and right-handed sugars and nucleic acids. And the question is why this happened and what was the driving force for this problem? And this prevalence of certain handedness of molecules in biology is called biological homochirality. And today we will talk about this problem. And why homochirality is necessary and why it is important for origin of life? Well, first of all, like life and the origin of life is heavily dependent on getting a genetic molecule which can self-replicate and transmit its genetic information. And replication of a genetic molecule like RNA, which both is in a chiral geometry and consists of chiral monomers like nucleotides, and its functionality heavily depends on its homochirality. A homochiral genetic code cannot replicate through Watson-Crick base pairing because the heterochiral pairs won't really match with each other. And this has actually been experimentally demonstrated fact that you cannot realize a functional RNA world or RNAs starting from a homochiral pool of nucleotides. So we cannot even get functional polymers, which can then evolve into a homochiral system. We really need to address this problem at the origin of life, at the prebiotic chemistry state, where we are building the polymers from a pool of monomers. So we can safely say that without homochirality, we cannot realize an RNA world or like a functional genetic code which can then evolve into modern life. And this is actually the homochirality problem, and what was the driving force between this was listed in Science magazine as one of the biggest questions in science, as actually one of the biggest 125 questions. like what is the origin of homochirality in nature, like what kind of physical force bias biological systems and and gave rise to the signature of life on Earth. And it is convenient to break the problem in separate parts because it is actually a big problem, and it can be considered as like several steps. So first of all, the abiotic chemical synthesis is symmetrical, meaning it produces chiral molecules in equal amounts in a so-called racemic mixture. So you start from a racemic mixture of whatever chiral molecule you make. And then to address homochirality, you need to basically couple this symmetrical system to an agent that breaks this symmetry, such that it introduces a bias toward a direction, L or D, and you get a slight excess of one-handedness. And then you can amplify this excess by chemical or physical means to homochirality to realize, a homochiral compound. But this is not enough because life cannot start with one compound, and there are many chiral molecules that are relevant for the origin of life and periodic chemistry. So you need to be able to propagate that attained chirality to other molecules that are important and for the origin of life to realize a homochiral network. Okay. So essentially, the problem consists of multiple parts about breaking the symmetry, amplifying it, and propagating it to the entire system. So now in this talk, I'll talk about a mechanism. Honestly, the colors can't see colors. Am I colorblind? Okay, I guess, huh?
That's colorful.
That's colorful, yes. Like this is colorful as well. Okay. Do I have a laser pointer or no? Or a spear? Oh, there it is.
Yeah, there it is.
Wow. Are you sure this is the right way? This is heavy. Okay. Well, I'll try to use this. Has anyone done this before? Okay, so in this talk, I'll talk about a symmetry breaking mechanism and then a way of amplifying the attained symmetry, the broken symmetry, and then talk about a mechanism which can propagate the chirality from one compound to the entire network. So the symmetry breaking will come from an effect discovered in more physics research named Chirality-induced spin selectivity about 10 years ago. And the seminal experiment is the following one. So the researchers took a gold substrate, which is not a ferromagnetic substrate, and then put DNA monolayer on it, which is a homochiral monolayer of single-handedness. And then they shine light on the surface and then ejected electrons that are unpolarized. And they're unpolarized because gold is not ferromagnetic. And then they measured the spin polarization of these electrons after they go through this chiral monolayer. Okay. The interesting thing is, although when they're beginning this experiment, there is no bias in the spin polarization, after they go through this chiral monolayer, they get heavily filtered based on their spin alignment. And the direction of electron spin, meaning the electrons that can make it, the direction of their spin is dependent on the chirality of this layer. For instance, in this case, the downspin electrons can effectively go through. And when you flip the chirality of the monolayer, for instance, from right-handed DNA molecules to left-handed oligopeptides, the other spin state now gets effectively filtered out. So there is some chirality-dependent coupling of electron spin and molecular chirality. Okay. And this is a room temperature experiment, and it's a strong spin polarization. They got spin polarizations up to like 60%. And this is the effect we will utilize. And another consequence of this effect is basically, when you polarize a molecule, a chiral molecule, meaning charge polarize a molecule, you realize like two charge dipoles, right? A charge dipole which consists of an absence of an electron cloud and an excess of an electron cloud. And when you polarize a chiral molecule, what you do is you basically push charge from one place to the other in a chiral system. So essentially, you do something very similar to the first experiment that I showed. When you polarize an electron, meaning you basically push charge in a chiral system in a way that it is filtered based on its spin alignments. So in this system, for instance, you see two chiral molecules that are polarized. And in the left-handed version and right-handed version, we have the same charge poles associated with, different ends of the molecule. And because these molecules have opposite handedness, the effectively propagated spin state is different. In the left-handed case, it is upspin. In the right-handed case, it's downspin. So essentially, when you charge polarize these chiral molecules, you also realize spin polarization. And that is the important thing that you need to remember. And it is like basically at the core of what I'm going to talk today. So, I mean, the summary, or like, I guess the takeaway message is electron transport through chiral medium is spin selective because of the CISS effect. And when you realize charge polarization, these closed shell chiral molecules become magnetic, okay? And we will utilize that. So, well, how did we utilize this in our research? So the CISS effect is about how chiral molecules filter out spin, right? But that's not what we exactly want to do. We kind of want to do the opposite. We kind of want to, like, instead of filtering out spin, we kind of want to filter out chirality because we want to bias chirality. So we can utilize this interaction in reverse, and basically using a spin polarization, we can bias chirality in one direction or the other. And where can we find spin polarization, meaning net magnetization? Well, in magnetic minerals. So now we are going to use magnetic surfaces with net spin polarization as chiral agents because of the CISS effect to bias molecular chirality in one direction. So now look at the system. We have a magnetic surface, and its electrons align, and we have a racemic system of left and right-handed molecules, right, in equal amounts. And as these molecules approach this surface, they basically get inductively charged polarized. It's a transient polarization due to like van der Waals interactions. And the charge polarization that two molecules have is agnostic to chirality, meaning it will be identical for left and right-handed molecules. But what is not agnostic to chirality is spin polarization, right? So just as we talked before, the charge polarization attained in these molecules will get accompanied by spin polarization because of the CISS effect. And now we will have different spin poles associated with the charge poles for these molecules. And by example, on the right-handed case, at the side that the molecule is approaching, the spin is downspin, and in the other case, it should be the upspin because these are opposite chiralities. So now there are, I guess, three steps, right? The first thing is the charge polarization. And then the second thing is the spin polarization that accompanies this charge polarization. And then the third thing is because now we have different spin states at these, different ends of the molecule, these molecules now will selectively interact with the spin polarized surface. And you can think of this intuitively as like, magnets, the same pole pushes and the opposite poles attract each other. So a similar effect will occur at the surface. Like in the case of shown here, right, in the right-handed molecule, the singlet-like up-down interaction will be favorable energetically. So that molecule will be pulled to the surface. And then the other one will be energetically unfavorable, and it will be pushed away from the surface. So essentially, any surface interaction that you do on a magnetized surface with chiral molecules will favor one chirality or the other, dependent on the magnetization direction. And this is not a magnetic field effect, okay? That's important to appreciate because magnetic field effects are very weak. You need to realize a high strength of magnetic field, but this is a spin effect. And the energy difference between left and right-handed configurations is the energy difference between triplet and singlet configurations. And that is much larger than room temperature. And in photochemistry, people are familiar with this, right? In the singlet and triplet energy gaps. And it's on order of like 100 millieVs, which is much, much stronger than magnetic interactions. That's why you can observe this effect at room temperature. So this was actually probably the most important bit or the most physics heavy bit of my talk. And then the rest of my talk will follow the ideas utilizing this, okay? So the next thing that I'll talk about, so this was about the symmetry breaking part, and I will get to the specific experiment in a second. But the next part was right after the symmetry breaking is the amplification part. Like how do we propagate the bias that we attain? So this is a short range surface interaction that is happening at the magnetic surface. And it won't really go beyond away from the surface for long distances. So what is the way to propagate this attained bias at the surface, which kind of the surface seeds the formation of one enantiomer faster than the other, but how are we going to make this better? So now we'll take a look at the crystallization properties of chiral molecules. And generally speaking, chiral molecules crystallize from their racemic solutions in two different ways. And in one way, I'm not sure if you can see blue and red balls, but they denote like opposite endedness. And in the more commonly, chiral molecules crystallize as a racemate, meaning a chunk of a crystal contains a homogeneous mixture of left and right-handed enantiomers. And a less common way of crystallization, about 10% of chiral molecules that crystallize do this, is the so-called conglomerate. And in the conglomerate, there's a net energy difference in the crystals, phase of, putting one chirality over the other, so that like individual seeds are enantiopure. But the overall mixture of different chunks, of course, is racemic. So as you can tell, the right-handed way, the conglomerate crystallization is a better way of propagating chirality, because once you push the system at the surface toward one direction, the crystallization basically will attract only the same kind in a way that it propagates the enrichment that is seated at the surface. So now the rule of the game is to select a molecule that crystallizes as a conglomerate and crystallizes on a magnetized surface. And hopefully, this molecule will be an important one. So now to go to the specific experiment, I'll talk just slightly about prebiotic chemistry and why we chose the molecule that we chose. So here you see a network, and believe me, it looks as scary to me as it may look to you. I'm not a chemist, but this is a prebiotic network that was mostly discovered by the group of John Sutherland. And it's a network that utilizes cyanide and electrons that are ejected from like ferrocyanide by UV irradiation to synthesize important precursors of life, such as amino acids, nucleotides, and lipids. And in this network, you start being chiral quickly after you start making sugars. And here you see the first chiral sugar, glyceraldehyde. And then that sugar basically reacts with a molecule that you can synthesize from glycolaldehyde to make a family of molecules called pentose-aminooxazolines. And then these are direct precursors of nucleotides. And they make cytidine and uridine, the pyrimidines, in very high yielding steps. And essentially, at that step, there's a molecule in this family of molecules called ARIA, ribose-aminooxazoline, which is the right stereoisomer for making ribonucleotides. It's the ribose-like sugar one. And it's a molecule that is pretty insoluble, so it crystallizes in water solutions. And it doesn't racemize in solution. So essentially, if you attain this molecule in its chiral form, it doesn't, lose its chirality. And moreover, it crystallizes as a conglomerate, which is not a very common thing. And it's a direct precursor of nucleotides, okay? So once you make a simple sugar and then make this family of molecules and get to the level of RAO, if you can somehow get this molecule in a homochiral form, you basically get your nucleotides in a homochiral form. Because this molecule is a direct precursor of RNA, and that is probably the most important thing. And the fact that it crystallizes as a conglomerate, which is a rare crystallization property. So it seems like an ideal molecule to work with. So now we took this molecule from its racemic solution, okay? And then put that on a surface that is magnetized. And the surface we use is magnetite. It's a type of iron oxide, which is the most commonly found magnetic mineral on Earth. And it's also the most magnetic mineral, natural magnetic mineral on Earth. So like, the both arrows point to the same thing. And we crystallize this molecule on a magnetite surface and get its crystals, and then collected individual crystals and then analyze their chiral properties. So on the left, you see, crystals of RAO on a magnetite surface. And then on the right, you see the circular dichroism spectra. And circular dichroism is a way of measuring chirality of molecules in an optical way. And a signal, positive or negative, indicates the presence of enantiomeric excess, either toward right or left. So what you see here is, dependent on the spin direction, we either got right-handed or left-handed enantiomer of RAO, although we started from a purely racemic mixture. And the excesses we got reach up to like 60% enantiomeric excess. And this is a really high number if you basically compare this to the excesses that people get from experiments that utilize like cosmic rays or circularly polarized light or certain like other effects. Like people were talking about like 0.1% EE, and if that is even, an authentic measurement. So this is like a really, really strong enantiomeric excess that you get from a purely physical effect, starting from a purely racemic mixture. And the black line, the central line you see here, is a control experiment. And in this experiment, what we did is we did this thing, but on a silicon surface in the presence of a magnetic field. And silicon is not a magnetic or magnetizable mineral. So essentially, although there was magnetic field present, because there was no net spin alignment, we didn't see the effect in silicon, which indicates that this is a spin effect as opposed to a magnetic field effect. And then you can repeat this experiment and basically get to a level of, what is the average EE enantiomeric excess you get in these experiments. And we got on average about like 40% EE over like different runs. And then also did an experiment in which like when we crystallize a solution which already contained excess from the previous crystallization, and then tried to find the lowest excess about which we can get to homochirality in the subsequent crystallization step. And we found that like around about 25% EEs, if you recrystallize a solution on the magnetite surface, you can go and boost the system to homochirality. And that is already below the average EEs we get, which is around 40%. So essentially we say homochirality in 2 crystallization steps because of this reason. So you basically crystallize once, and then after you dissolve and crystallize again, you boost the system to homochirality. So now I will talk about the another manifestation of this effect, which is also related to realizing highly spin polarized surfaces on early Earth. And the motivation for doing this is, first of all, it is happening probably whether you want it or not. And the second thing is the Earth's bias that, or the Earth's magnetic field and that the resultant magnetization because of that field is going to be statistically significant but not uniform. So how can we make this better on a natural scenario? So now I'll talk about how chiral molecules can be utilized to magnetize surfaces as well, again using the same effect. So this is no different than what I showed before. It is just another way of looking at things. So here we have a surface that has no net spin alignment to begin with. So, you know, an equal amount of up and down arrows, I guess. And then the solution is now homochiral as opposed to being racemic. So now as this molecule approaches the surface, the same thing happens. It gets polarized, charge polarized, and then spin polarization accompanies charge polarization. And as this molecule approaches the surface, now again, because of the preferential energetic bias towards up-down configurations, this spin now polarizes the domains underneath itself. So essentially, chiral molecules, just as magnetic surfaces can bias chiral molecules too can magnetize magnetizable surfaces. Okay, so there is like kind of a reciprocal nature of the effect which can be utilized like a feedback and can be used to basically polarize surfaces just using chiral molecules. So we measured this effect using a technique called magneto-optical effect microscopy. And the details of the measurement don't matter that much, but here you see a surface of a magnetic surface on which chiral molecules are crystallized in that like white box. And then as you kind of go to a regime where you can see the image contrast between these two areas, you basically see a different shades of color in an area that is like nearby around the crystals, and in the other area is at a different like magnetization contrast, which indicates that the chiral molecules essentially like pin the domains underneath them and magnetize the domains around themselves. So essentially like chiral molecules act like little magnets or like little magnetic dipoles and bias the domains underneath and around them. And it's almost like an avalanche effect, because once you crystallize chiral molecules and bias the domains around them, the magnetic interactions in the material itself basically favors the same direction. So the chiral molecule comes, it magnetizes the domains underneath them, and the domains now will favor the same spin direction for the nearby ones. So then that affects the nearby domain and magnetizes that as well. So it is kind of like an avalanche magnetization that it can be triggered by chiral molecules. So here you can see that actually the magnetization spreads around an area that is larger than the area that is covered by the crystals. So it's a nonlinear effect. And there is like a little bit something more interesting about this. And here we did a hysteresis measurement, basically looked at the magnetic properties of the surface with and without chiral molecules. And this is a hysteresis curve. And you can read this kind of starting from here. And we look at two different areas. The blue area here is an area that is far away from chiral molecules. And here, the area is nearby chiral molecules, the red area. And here, we apply magnetic field, and it's a strong negative magnetic field, and it magnetizes all the domains in the kind of negative direction, and they're in the same length. And then you start increasing the field in the opposite direction. So you move from here all the way to here, okay? And then here, because now the opposite direction magnetic field is strong, these domains kind of give up, the blue ones that are far away, the free ones. And then they magnetize in the direction of the applied positive magnetic field. But notice here, the domains that are around the chiral crystals do not give up as easily, because the added magnetic field strength, effective added magnetic field strength by chiral molecules. You need to apply almost like 1 millitesla more field to beat the domains nearby the chiral crystals as well. And then they, of course, give up as well. And then you can also read this in the opposite direction, which gives the same physics. And the added magnetic strength by the chiral crystals is about like 20 times stronger than Earth's magnetic field. So essentially, if anything changes in the environment, like Earth's magnetic field flips, or if there are any thermal fluctuations, the induced magnetization at the surface will not flip because it's protected by this factor that is added by the chiral crystals. So essentially, you realize a persistent magnetization that is stable against the fluctuations in the environment. And then this ensures that the same chiral bias will be present in the environment no matter what. And also it will ensure that there will be like a uniform magnetization bias at the surface. And that's where it matters. So you can easily see that this effect can be kind of considered in a feedback, right? The effect that I talked first is the enantioselective crystallization. The effect I talk afterwards is chirality-induced magnetization. And of course, you can start from a completely racemic system, which doesn't involve homochiral molecules or fully spin polarized surfaces, but you can utilize this effect in kind of together. So essentially, if one enantiomer of RAO just stochastically starts crystallizing on an area, because of effect number 2, that will magnetize the domains underneath itself. And then, because now these domains are aligned, from the racemic solution, they will attract the right-handed enantiomer faster because of effect number 1. So this thing can basically spontaneously break the symmetry and work as like a cooperative feedback. And of course, you don't have a fully stochastic system because you have a statistically significant alignment that comes from Earth's field. So if you realize this effect for like on a large region, you will have more right-handed than the left-handed one. So this effect can be considered in a feedback. And I just put this in like kind of together here. You know, you have a natural magnetization from Earth's field, and then you have a system of racemic ribo-aminooxazolines, and then you first get the crystallization of enantio-enriched RAO, and then when you recrystallize, you get nearly enantio-pure RAO. And then as these crystals grow, they basically align the domains underneath them, and then you can do subsequent crystallizations because prebiotic chemistry goes on. So this effect can reinforce itself. So far, I guess I used 40 minutes?
35.
35? Oh, even better. Okay, so now I will talk the last part about my talk. So now I talked about so far, I got tired carrying this. Oh, it's so heavy. So now, until now, I talked about realizing chirality in a single compound, right? But I promised in the beginning of my talk that chirality problem is not restricted to making one compound homochiral. And it must be spread. So now I'll talk about what is the effective way of propagating chirality in a prebiotic network. And I will like talk about different classes of molecules, in particular nucleic acids, peptides, and metabolites, which are all relevant for life. And I will ask this question, like forget about the symmetry breaking mechanism, forget about the amplification. Which molecular class, peptides, nucleic acids, or metabolites would be ideal to start chirality such that we can propagate it? So now we will forget everything we talked about the assumptions, you know, like how can we get homochirality? We will just ask the effective path of propagating chirality. So let's start from a metabolic cycle like the Krebs cycle. So not all molecules in the Krebs cycle are chiral, but some of them are chiral. And for instance, malate is chiral, isostrate is chiral. And tartrate is a chiral molecule. In fact, it's the molecule where chirality was discovered by Pasteur, tartaric acid. And tartrate can be reduced to make malate. And tartrate can crystallize as a conglomerate. So in principle, we can get this molecule homochiral. And then reduce this molecule to make malate homochiral. But then what? Okay. So the problem starts here. Let's say you can start chirality and establish chirality in this cycle. Then what? Malate either oxidizes to oxaloacetate or dehydrates to fumarate. Both of them are achiral. The same thing for isocitrates. I don't have a direct path of making this one homochiral, but let's say we inject it with some mechanism. Again, it is surrounded by achiral molecules. So essentially, even if you establish chirality in a metabolic cycle, you lose it because in the absence of chiral enzymes that drive these processes, Krebs cycle is a chirality grinder. You put it and then it just like gets lost because of achiral branch points. So essentially, again, we forget about what we talked about before. Even if you can start chirality in a metabolic cycle, you cannot propagate it within or through and after metabolites. So metabolites don't seem like an ideal place to start chirality. Well, what else? Amino acids. So amino acids are plenty, right? There are like 19 chiral proteinogenic amino acids. And they have idiosyncratic chemistries. And also, they have like different dipolar alignments, meaning like, although all amino acids are left-handed, they don't all interact with chiral agents in the same way. Like they rotate polarized light in one direction or the other, depending on what amino acids we're talking about. So their interaction with chirality is a little bit idiosyncratic, specific to a certain like molecule. And if you talk about the R mechanism that like we just discussed, and if you ask the question, what amino acids crystallize as conglomerates, there are three of them: glutamic acid, threonine, and asparagine. Well, they crystallize the conglomerates, but they're extremely soluble, so it is very hard to crystallize them to begin with. So you need like a large concentration of them. And also you need to crystallize every single of them in the common direction. And that is not guaranteed because again, they can attach to the surfaces in different directions because they have different functional groups. So essentially, it is not as bad as the problem in the metabolites, which is the chirality grinder, but it seems more like a tedious task to resolve every single amino acid in a certain direction. Unless there's a better way of doing it, okay? Unless there's a common way of making peptides homochiral as well. And that can be done using the genetic code. And look at this result by the Sutherland group, and there are actually other results by Sutherland group and Shostak group that follow this. And here what they do is they make coded peptide synthesis, in particular the amino acylation step, like charging the RNA with amino acids, using homochiral RNAs, starting from a racemic pool of amino acids. And here what they do is they look at the stereoselectivity of peptide formation, starting from a racemic pool of amino acids. And what they see is if you use right-handed RNAs to make peptides, left-handed peptides form almost 10 times faster than right-handed peptides. And they tried this with alanine, lysine, leucine, and valine, and it all goes the same way. So it is not idiosyncratic. And moreover, the chirality of it is actually controlled by the sugar's chirality. So when they flip the chirality of tRNA, this is an unpublished result, but it's on its way, and utilize left-handed RNA, now right-handed peptides form 10 times faster. So essentially, you don't have to go through every single amino acid. You just do is you make the genetic code, which kind of is a dynamo of making peptides, form. And then even though you start from a racemic pool of amino acids, you make peptides selectively using RNA, which I think is a much more elegant way. So if tRNA can make L peptides and bias them to form faster, then eventually L peptides will make L enzymes, and an enzyme catalyzed metabolism will of course now be homochiral, because now you don't rely on the inherent interactions, but enzyme catalysis with homochiral enzymes. So although you cannot propagate chirality from metabolites to amino acids or nucleic acids, you can do the other way around. You can propagate chirality from enzymes to metabolites. So the direction of chiral information is more feasible if you go from peptides to metabolites. Of course, this is a long step of evolution, right? I'm not claiming that these are individually solved steps, but for the purpose of chiral information, we can say this. Well, okay, then if we agree with this, well, what is the way of making tRNA homochiral or RNA homochiral? If you make individual constituents of it homochiral, meaning ribonucleotides, then you can get your genetic code homochiral. And if there is a precursor, such as RAO, then making this compound homochiral, you can propagate that first nucleotides into RNA, and then with stereoselective amino acylation to peptides, and then to enzyme catalysis to metabolites, which is a much more effective way of propagating chirality. And I call this the central dogma of homochirality because it actually resembles the flow of sequential information, which is established by Francis Crick in the central dogma, which basically says that the information flows from nucleic acids to peptides, but not the other way around. And in the similar spirit, the information, the chiral information from flows from RNA to peptides and then to metabolites, but not the other way around. So essentially, we can reduce the colossal problem of homochirality of making every single probiotic compound homochiral is to make RNA homochiral from a single shared precursor of ribonucleotides, from which then chirality can propagate. And we know an effective way of doing it. So this is the end of my talk, and I will just summarize it now. And homochirality is a feature of life on Earth, and its origins are likely prebiotic because without homochirality, we cannot get life started. Oh. And magnetized surfaces are environmentally plausible agents that can break the chiral molecular symmetry due to the CISS effect. And RAO is a ribonucleotide precursor, a central one, that can be obtained in its homochiral form through its crystallization on a magnetite surface. And chiral molecules like RAO can magnetize magnetic surfaces and thus amplify a weak natural magnetization of minerals. And basically, this effect can be realized in an environmental setting as well. And finally, homochirality obtained at RAO can be effectively propagated to RNA and then to peptides, and then ultimately through enzyme catalysis to metabolites. So we can address the whole network chirality. Well, I, of course, went through our results kind of quickly, but if you want to check out the papers, here's a set of my PhD work, which you can look at. And the last bit I didn't talk about for time constraints, but if you have questions, ask me about it. I can quickly say what they are. So in the geochemistry part, like I mostly worked on the formation of magnetite from like, you know, in like lacustrine-like conditions, and looked at first how magnetite forms in prebiotic chemical pathways and what are its magnetic properties. And the second paper I looked at in a natural lake setting, like, you know, in a modern lake, how much bias that Earth gives us from Earth's magnetic field. And in the final paper, I looked all of these effects in varying temperatures and see like how does temperature affect these things and if this is robust at different temperatures or not. So I'll end here. I'll thank my PhD advisor, Dimitar Sasalo, who you see us in Pilbara. I'm dressed like David here with the funny head and and well, not today. And also my collaborators, probiotic chemistry, John Sutherland and CISS effect, like my collaborators in Israel and other geochemistry people from Nick Tosca and Rich Harrison, Roger, Cambridge and Harvard. So thank you.
Questions? Okay, Kevin, let's start with you.
So why does a L enzyme necessarily need like mostly R metabolites? Because I mean, like enzymes can break down L amino acids, they can break down D sugars. So why does it necessarily need homochirality?
Well, I guess like there I kind of skip a lot of steps in that, like in modern biology, like L enzymes control the metabolic activities, right? And then they make, let's say, L malate and then D-isocitrate using the same enzyme. Of course, this stereoselective interaction is very idiosyncratic and it's not guaranteed. But in that like step, right, we assume that that system evolves into the system that we have now in modern biology. And we know that that's the way it does it.
So do you think at all like the metabolic had to be made to be homochiral separately, if that makes sense?
Well, it can be made homochiral, but it cannot propagate its chirality to other systems, which I guess first, because
which you formed all of your homochiral RNA,
Mhm,
and then you start making its opposite chirality peptides.
mhm,
If they're both existing in the same space and they're still sort of relying on that magnification of the magnetic field, aren't they going to start destabilizing that? If you've got a bunch of L and now it's making a bunch of D, like they are going to have opposite effects on the minerals around them that are sort of stabilizing that system. So how do you deal with that?
I see. Yeah. So first of all, like chiral molecules and magnetic surfaces, when they're just in the present in the same environment, they just don't interact the way that REO interacts with it. You know, you need to realize a surface effect, and the thing has to crystallize as a conglomerate to basically kind of disrupt your surface magnetization. Like again, this is not a magnetic field effect and it occurs at the surface, and you need to, for instance, crystallize an amino acid on that surface to disrupt the magnetization in the other direction. And it's a good question that, like, you know, if that would happen, like I would say probably there are two things to consider. First of all, the amino acids are very soluble, the ones that crystallize as conglomerates. So I think you need about like 6 molars to crystallize asparagine, for instance, in water, which I don't see as very plausible. And the second thing is I also do not like see these things happening in the same exact small location, because, right, like you make REO, let's say using a location of the lake, which is covered by magnetite, and then you make RNA out of it, which requires several steps. And then to get to the level of making peptides, you probably like need to like separate your system from that local environment, you know? So you're right, if what's happening all in the same environment, and if you're able to like crystallize these things, which is very unlikely, you may do that. But other than that, they don't really interact with the surface the same way that REO does. Yeah.
Roy?
So since you're looking at lakes now, what is the crystallization phenomenon sensitive to the solution chemistry of the lake? What else is dissolved in the lake, pH and so forth?
Yeah, so that's actually this paper, which hopefully will come this year. Let me see. Yeah, with Google Whitson, who is working with John Sutherland. So in that paper, they built the third one, I guess, here. It's heavy, right? I told you. So the thing is, like, I studied the crystallization of REO in the presence of molecules that resemble REO, because those are the likely ones to incorporate themselves into the crystal lattice and disrupt or change or not do anything to the crystallization of REO. And other pentose-aminooxazolines, which are produced in the same way as REO. In particular, one of them actually gets embedded in the crystal lattice and changes how RAO crystallizes. And because of that, they use the differential solubility difference of that molecule from RAO to basically wash away first that, because if RAO is much less soluble than that molecule, when you do a crystallization step, you get stir-selective cleaning because the other one just leaves the solution, right? And that was like the solution, but that was kind of a suboptimal solution because these are in the same environment, really. In this paper, what we found is as opposed to making the solution pure, we made it more complicated and hope that when we make it more complicated, the interactions of the molecules with themselves will nullify that effect that molecule is doing, okay? So when we put other molecules, meaning products of, for instance, glycolaldehyde that we make from the glycolaldehyde, we found that the crystallization behaves much more favorably to our needs than the case of the more pure solution. And I think that is the whole mark of prebiotic chemistry, right? Like if you find the right chemistry, making things more complicated will make it better. If you find the wrong chemistry, making things more complicated will make it even unclear. So that was an interesting result. Of course, this was with molecules that resemble RAO, thinking that those are the most likely ones that would affect this crystallization. But for instance, a next step that we want to do is with David maybe is try this with phosphates, right? Because we now know, like Sebastian's work, with like lakes that have a lot of phosphate, that also carbonate. And in those lakes, you need a lot of carbonate to get a lot of phosphate. And actually these lakes also must have phosphate because you make nucleosides, but then to get to the level of nucleotides, you need to couple them with phosphate. And, I would like to check, for instance, how this thing changes with phosphate and carbonate in the solution.
Sure, could just take some of their water. We've got some.
For instance. Yeah.
Tom.
So, it's fascinating, but so you need the magnetite
Yeah.
and so 200 nanometer photons.
Say it again.
So you need that, RGB photons.
200 nanometer photons.
Why?
Polarization.
Oh, the interaction that relates to the CISS effect is a dark thing. It doesn't require any UV radiation. It's completely dark. I mean,
You got this nice plot.
oh, that's just for analytics.
That's just a measurement, right?
That's just a measure. Like, you mean this?
That's just.
Oh, that's just a measurement.
That's the measurement of the enantiomeric excess.
Okay.
Yeah.
But that's, yeah, basically utilize it like optical transition of these molecules with polarized light to measure how much EE they have. But this is a dark process.
Show me the vision how this happens on an early Earth
Yeah.
that just happens to be a water world. There's no landscape, living covered with water.
Yeah, but-
And it's not an oxidized world either. So it's hard to produce magnetite.
Well, I mean, first,
Magnetite's common on Earth today, but it wasn't probably earlier.
like, first of all, I don't think like there is a requirement that the Earth was completely like a water world because you can.
It's a possibility.
It's a possibility, but like there's nothing that is against of making, for instance, things like Hawaii or Iceland
Yeah,
from.
it was likely because Earth had no water originally.
Yeah,
All the planets did. Yeah, and so, it's certainly a possibility. And Hawaii's don't last very, in the last couple of million years.
yeah, but I mean, for prebiotic chemistry, like, do you really need that much time? It's chemical timescales.
I don't know, but anyway, I was just wondering, like, in the early Earth was quite different than it is now.
Oh, for sure.
Certainly you want very fine grained magnetite, right?
You need single domain magnetite.
Yeah. Super magnetics.
Not that, but yeah, I mean, so the thing is, first of all, you cannot have oxygen because if you have oxygen, you make hematite instead of magnetite. So anoxic works for you. And we study this under anoxic conditions for that reason.
But you make the magnetite by photooxidation. as
But
one mechanism.
That's one mechanism, but photooxidation just accelerates the oxidation. But you can also get like chemical oxidation, for instance, with bicarbonate carbonates. That also gives you that. So like there are ways of oxidizing iron chemically, and you have to do that under anoxic conditions, because if there's oxygen gas, that actually makes hematite instead of magnetite. And actually, like that's one of the papers that is unpublished. We studied magnetite production under earliest conditions and studied its magnetic domain properties and looked at what are the sizes of the particles we make. And mostly you make single domain magnetite crystals in water.
I mean, there's plenty of magnetite in Archean ion formations when there wasn't any oxygen.
Right.
I mean, of course, biology can be involved, but even without biology, there are experiments in the literature where you can make magnetite in anoxic environments.
Right. Also in surface hydrothermal systems, like rocks like ophiolites have a lot of magnetite from, you know, iron oxidation again. And like those environments, I guess, resemble more like Iceland, right? So yeah, I mean, magnetite was discovered in zircons, in Mars, in everywhere.
Any more questions? Sebastian?
Maybe you said it, so are metabolites also homochiral?
So metabolites have a lot of molecules, and actually mostly they are not chiral.
Oh,
Molecules
yeah,
are achiral molecules, but several of them are homochiral.
Yeah, I was just wondering with the Krebs cycle chirality grind, as you said, like,
Right.
so if you sample like my body right now, like would metabolites be chiral? That could be chiral? And if so, how would that chime with your central dogma for chirality?
So
Like I would expect with your central dogma of chirality,
yeah,
for it to fizzle out at some point in the metabolite cycles where these grind points are, the branch points are that you mentioned.
right.
And
So
then actually-
why does it happen now? You're
Yeah.
saying, right? Because now we don't rely on inherent reactivity of molecules. We rely on enzyme catalysis. So like essentially like this, let's say dehydration of malate to fumarate is controlled by enzymes. And then the enzyme chirality affects the chirality of the product or the reagent, you know? So in the absence of enzyme catalysis, Krebs cycle is a chirality grinder, right? That's why like going from peptides onwards to enzymes, then to metabolites is a longer route, but it's the more feasible route to make metabolites homochiral. Yeah, great question.
Okay, one last
Roy?
question from Roy then.
Yeah, so is RAO, a precursor of the purines as well as the pyrimidines?
Yes, Roy hit the nail on the head.
Well, so the purine synthesis is an unsolved problem.
Yeah, I mean, the thing is, like RAO is a precursor of pyrimidines. And purine synthesis, I mean, beyond their homochirality is an open problem. We just don't know how to make them. And seems like it's not my problem, really.
Well,
It's more like John's problem
You
right
can
now.
make them, but in not high yield like you can the pyrimids.
Yeah, it is, but my feeling is that it's hopefully they will come from RAO, and John right now is working on it. You know, John is, because of these results, like he is like thinking that it should come from RAO, and he says that he made it, but I heard that I think many times. But even if it doesn't, okay, I think making half of the alphabet homochiral will probably bias it in a way, in the direction that, you know, to the other half. But of course, you will lose a lot of material doing that. But it's of course not as bad as starting from a completely symmetrical system. But hopefully it will come from RAO and we will have no problems. Yeah, but question.
Okay, well, we're over the hour. So one thing I forgot in my introduction is that Furkan's thesis won something called the Gold Harbor Prize, which is the best thesis at Harvard in experimental physics. So congratulations and thanks for your talk.